So pleased and so delighted. I welcome you to our today's class in biology. And I've just picked a paper that I have found in my archives. This is a paper that was done by the students in our country in the last year's final examinations in as far as our O level is concerned. And the questions brought here are picked from the coursework that the students were exposed to throughout their studies. I'll move to the first question. And the question says, the following diagram shows an apparatus used in ecological studies. And looking at this diagram, what comes into the mind of the student is that this question must have been extracted from a particular concept that was introduced to the students in the first topic called Introduction to Biology. And in that topic, the students are always exposed to several apparatus that are normally used to collect specimens in the environment during a particular study. And some of these apparatus, apart from the one we've actually been given here, are what we call a sweep net. And you must have been told that a sweep net is used to collect flying insects. So it has a handle and a netting or a sack-like net that normally picks the insects flying, like the house flies, and of course the mosquitoes and any other insect that have the ability to fly in the air. So any flying insect in the environment can get collected by a sweep net. So that becomes the first apparatus for us to mention. And appropriately with a handle for the user to be able to use the apparatus to collect the insects. And an appropriate depth for what we have in our netting section of that apparatus so that you can be able to have the insects contained after they are collected from the environment. And apart from this, you'll always be able to find what you call a pitfall trap. So any small crawling animals on our earth's surface can fall in the pit. So it just has to have a particular arrangement where there is a pit or a hole that gets dug in the ground with a dustbin cover that can prevent entry of water into this pit, because when the pit gets filled with water, it would be very difficult for the scientist to collect the small crawling animals on the ground. So the dustbin cover prevents entry of water into this pit that collects these small crawling animals. And this dustbin cover is normally supported by some structures to just ensure that it actually gets erected from the ground. So collect small crawling animals from the ground. And we have what we call a specimen bottle. And the work of the specimen bottle is to ensure that once the specimens have been collected by this apparatus, they get kept into these specimen bottles. And the specimen bottles can actually get well aerated to prevent the dying of these specimens if the specimens are needed when alive. For example, if I've collected some cockroaches from the environment and want them to be alive so that I can be able to monitor their movement and see how they can be able to actually use their limbs to move around, then I'll actually have to ensure that where I'm placing them in that specimen bottle has to get well aerated so that these specimens do not end up dying. So the specimens can get kept aerated or airtight with a lid that completely closes them to ensure that no air enters or no air exits. And apart from this, we have what we call the forceps. Forceps are very good at collecting specimens that are poisonous or have the ability to sting, and very mobile or very injurious specimens can also get collected using a forceps. It has to have that appearance of what you call a pair of scissors, so that from that rear end, the user has to actually hold that forceps and pick up that specimen that is so poisonous, injurious, or can be able to sting the user. So for any injurious, poisonous, or stinging organisms, then you can decide to use what you call a forceps to pick up these organisms and keep them very safe for the purpose of the studies intended to actually occur and prevent injury to the user or any form of poisoning or damage or injury to the user that actually has to handle these organisms. We also talk about what you call a putter, and this is a putter. So name the apparatus. This is the putter, and the work of the putter is to suck small animals from the backs of the trees and rock surfaces. That's why you can look at this arrow in this. So this is the tube for suction. That means the user is here to suck. And look at this arrow. It actually points towards the entry pathway into this apparatus. So that means this is the tube for collecting the specimens, the tube for collecting the insects, the tube for collecting the small animals. And this is the tube where suction is applied so that those organisms can get sucked from the rock surfaces and backs of trees. So tube for applying suction, tube for collecting the organisms that are supposed to get collected. And once suction is applied here, the organisms get sucked from the backs of trees or rock surfaces. They fall into this container-like apparatus. And we've said that there's what we call a mosquito netting or a wire mesh that just ensures that no dirt after the sucking enters the mouth of the user. Because there's a possibility that when this tube for collecting insects gets inserted into the back of a tree or a rock surface, then that means dust or any other materials like the small leaves and whatever may actually get found into that environment can find it into the way of the user's mouth. So that means you're supposed to ensure that this part is what we call a mosquito netting to prevent the entry of what? Dirt into the mouth of the user. So to prevent dirt entering into the mouth of the user, that's the work of the mosquito netting or the wire mesh, or what we call a polythene-like paper that is well able to allow air to enter so that the sucking can occur very well to suck the organisms. So, and of course, this container must be made of what you call glass. Glass is transparent. If you look at physics, you're told we have three main types of materials: opaque materials that do not allow light to pass through them, and again, you cannot be able to see through them. You have what you call translucent materials. Light can pass through them, but you can't see through them. Then transparent materials that can possibly allow air and light, of course, to pass through them, and you cannot be able to actually say that you don't end up not being able to see through. So you can be able to see through that transparent material, and then light can pass through it. So this must be made of a transparent material like glass that can actually allow light to pass and as well be able to enable us see through, so that you can be able to see which organisms have been collected in this apparatus. So then the glass is used to make up this container for the purpose of visibility, to clearly allow us see which organisms have been collected in the container. So for visibility, for transparency, so that you can be able to see very well whatever is inside these organisms. Hello? Hello? Yes? Hello? Unajua si nimekwambia naenda kutafuta chakula.